Alright, so today is going to be about viruses and some of their basic characteristics. In the animation, we'll eventually talk about how viruses copy and make more of themselves, which is what this animation is showing. So we'll go through this a little bit later. So here we go. So the basic question of today is, what is a virus? And when you look at the definition of a virus is a biological particle composed of nucleic acid and protein. It's a very, very vague definition. In fact, all the definition really is just a list of two things that viruses are made from, nucleic acid and protein. So we don't really have a good definition as to what a virus is. They're a biological particle. They affect living organisms. They perform certain acts of biology, as we'll go into. What we also know about a virus is that they are also called intracellular parasites. The prefix intra, of course, means within. So these are parasites that must reside within a host cell. And we'll go over some of their other features, but a virus, in order to make more of itself, must gain entrance into a host cell. And once it gains entrance into a host, it is able to, like a parasite, live off of the host and, in this case, make more of itself. So right now there's kind of a debate. Are viruses even considered a living organism? And there's obviously two sides to this debate. So let's look at the arguments for yes, they are alive. Well, one argument for yes, they are alive is that they are able to make more of themselves. And this, of course, in biology is called reproduction. Now, do they reproduce like a typical cell, either sexually or asexually? Not at all. But they are still able to make copies of themselves, although there are restrictions according to how they make copies. Viruses have nucleic acid. Viruses have DNA, or viruses have RNA, and we know that living organisms in their cells have DNA and RNA, so this is another argument for that, yes, they are alive. Viruses can adapt to their surroundings. You know, bacteria can adapt to the overuse of antibiotics, and viruses are no different. Viruses are adapting to the overuse of antivirals. It's survival of the fittest. Some of the antiviral medications that people take for various sicknesses are becoming less effective because of natural selection. We're eliminating the weak, and the virus are able to make more of themselves. And another for the yes category is that viruses have organization to them. There are different parts, and each part does a separate job for the overall good of the organism. And this is something that we typically see in cells. Cells have organization to them, and they have a nucleus, and they have all these different parts to them, each of them doing different jobs. Now, I'm not saying that viruses have cells, but darn it, they do have organization to them. So are viruses alive? Let's look at the no debate. Well, no. One of the no arguments, they're not alive because they're not based on cells. And one thing biology has taught us, one of the basic principles of biology is the cell theory, that all life is made from cells. Well, we know viruses aren't. Now, science has been wrong before. We've had to amend and change definitions and redefine biology in the past. And so if we do consider viruses living, we will have to make some adjustments into some basic biological concepts. Like, for instance, the cell theory. All life is made from cells. Well, viruses are not made from cells, and if we ever do consider them a living, we'll have to make some amendments to our basic understanding. Another argument for viruses are not alive is that they can't reproduce on their own. They are able to make more of themselves, but the way that they do it, they require a host. And once they invade a host, they will then take over the machinery of the host, and the host actually makes more of the viruses. And we'll go over that life cycle later on in these notes. Another reason why some people consider viruses to not be alive is that they do not metabolize energy, which basically just means they don't feed. And this is kind of interesting because we know all cells, all life has basic needs. One of those needs being the need to feed. Whether it's an autotroph or a heterotroph, they still need organic molecules to break down to create into ATP and therefore use that ATP for cellular processes. Well, viruses don't. They're not made of cells, therefore they don't have the real need for ATP and they don't require energy. And that brings us to the next one. They don't perform cellular processes. There is no mitosis in viruses. They don't do meiosis either. Transcription and translation, well, we'll kind of debate whether or not viruses do them. Viruses will trick cells into doing transcription and translation, but the virus itself does not. So no basic cellular processes are completed. So right now, the official answer to are viruses alive is no, that they're not, because we know that there are 6 big, broad categories of life, 6 kingdoms of life. And there's 2 kingdoms of bacteria, that would be the archaea and the eubacteria. And then you have the 4 eukaryotic kingdoms, the protista, the fungi, the plant, and the animal. And in those 6 kingdoms, we don't place viruses in any of them. So currently, viruses are not considered a living organism, even though they do have certain features in common with life. So let's look at a few other virus parts, basic parts of a virus. All viruses have an outer coating called a capsid. Like it says in the definition, it's a coat of protein that surrounds nucleic acid. Well, now that we know that the capsid surrounds nucleic acid, the second part that they all have in common should be obvious. All viruses have nucleic acid. Viruses are kind of classified according to whether or not they have DNA in them or whether or not they have RNA. So viruses have one of these 2 types of nucleic acids, DNA or RNA. They do not have both. So for instance, HIV is a RNA virus. It only has RNA into it, or inside of it. In the picture we see here, this is a virus known as a phage, and if you look at what looks like little legs at the bottom of the virus, well, these are called tail fibers. And like it says, these are not used for walking. These are not legs. They aren't used for crawling. They're used for attachment purposes. So they're little spikes of protein, and they will attach themselves to a host cell, and we'll go into what happens after they attach in a little bit. And notice from the picture the shapes all vary. Some of them are round, some of them are helical, some of them are tube-like. So the shapes and sizes will vary. When it comes to their ability to replicate or to make more of themselves, well, we often study what are known as phages or bacteriophages. And bacteriophages are viruses. And the reason they are called bacteriophages is they're viruses that attack bacteria. And we often study these because of their simplicity. Bacteria are easy to study, they multiply quickly, and a lot of what we learn about bacteria viruses, we can translate that into animal and human and plant viruses as well. So we learned a lot about how viruses behave and how they work by studying bacteriophages. So the replication, how they make more of themselves, is very similar with animal viruses. Here we see in this picture dozens and dozens of phage viruses. Again, bacteriophages is another word for a phage. So in the picture you can see dozens of phage viruses attacking an E. coli bacteria cell. So there's one bacteria cell kind of under attack by dozens of viruses. And the way that these viruses replicate, again, is very similar with animal viruses. That's why we often study phages, bacteriophages. We're not going to give people a virus infection on purpose just to see what happens. There are ethical reasons, of course, why we should not be doing that. And so what we're going to show today is that viruses go through 2, and I hesitate to use the word life cycles because the word life implies that they're living, and as we just got done saying, viruses are not currently classified in one of the 6 kingdoms of life. But viruses go through 2 reproduction cycles. We're only going to talk about one of them today in these notes. We'll talk about the lytic cycle today, and we'll talk about the lysogenic cycle another time. So let's start with the lytic cycle. The lytic cycle can be broken down into a series of stages, and these animations are really good. Notice on the picture the virus is landing on these little bumps that are located on the outside of the cell. So the virus lands on the cell membrane, and you can see from the labeling of the picture, these little bumps are called receptors. And I like to use the analogy of a lock and a key. The receptor is kind of like the lock, and the virus like the key. If the key fits the receptor, we'll then proceed into step 2 of the Leydig cycle. So in this animation, repeatedly over and over, our virus is attaching to the receptors on the cell membrane. Once that happens, the virus acts like a key, and the virus will release some enzymes to weaken the cell membrane, and that will allow the virus to gain entrance. So we'll go into step 2 in just a moment. Actually, let's just go into it right now. Step 2 is called entry, and notice how the animation has changed. The virus is contracting, releasing its genome into the host cell, again the genome being the DNA or the RNA. So what happens is once the virus attaches to the receptors, enzymes from the virus will help to weaken the cell membrane to the point where the virus can just contract and penetrate its outer body, its capsid, and then inject its DNA or its RNA, its genome. So the DNA or the RNA enters the host cell. Here's another animation showing a way that viruses can enter. In this case, the virus landed on the receptors, and the cell swallowed it up through the process of phagocytosis. And so this is another way where viruses can gain entrance. So in all likelihood, the cell, probably thinking that this virus deserves to be inside, after all, it matches the receptors. It's kind of like it has a house key. So now that the virus has a house key, it matches the receptors. The cell swallows it in, probably thinking that it's some kind of organic molecule, some proteins that it might need for cellular purposes, and it will swallow up the cell, and once that happens, the DNA or the RNA from the virus is inside. Well, then we move on into step 3 of the Leydig cycle. Step 3 of the cycle is called replication, and like the name implies, replication implies that more copies are going to be made, and you can see that happening in the animation. The virus DNA, if it's a DNA virus, will be used to make proteins. If it's virus RNA, because again some viruses only have RNA in them, that RNA will be used as well to make proteins. So the DNA or the RNA of the virus will use the host cell's ribosomes to make virus proteins. Remember, ribosomes are a cell part whose job it is to make proteins. These proteins are going to be created through the process of transcription and translation. And I'm hoping that you recall these topics from earlier in the school year. Here's a little review of transcription here. Transcription is where DNA is converted into RNA. The difference here, it's the virus DNA is converted into virus RNA. That's through the process of transcription. After the viral RNA is created, the viral RNA will be converted into virus proteins. So the process of taking RNA and creating them into proteins, again, is called translation. And the virus does not have the ability to do these, but the cell does, the host cell. So this is why a virus needs to gain entrance into the host. The virus will use the ribosomes of the host to perform transcription and translation in order to make copies of itself. So this is step 3, replication. All these viral proteins are being created. Well, now that you create all these virus proteins, that leads us to step 4. Assembly. Those proteins are going to be assembled into new viruses. So what you see happening in the animation, again, you have 4, in this case, 4 viruses being assembled. In reality, it's more than 4. The animation is just showing that how the process continues from replication into assembly. And once the viruses have been assembled, we come to step 5, known as release. The viruses will release enzymes that cause the cell membrane to weaken, and eventually the cell membrane will lyse. And lyse is simply a word that means to burst. So in the animation, you can see that the cell membrane has burst, and those viruses are released. Well, keep in mind this is called the Lytic cycle for a reason. Once the viruses are released, the cycle will repeat itself. Here's a picture that was earlier in the presentation, and this is a picture of a bacteria cell bursting, and from the inside you can see dozens of little new viruses being released into this particular environment. All these viruses can then find a new host to infect. So that's a little summary of the Lytic cycle. And here we have on one slide all stages side by side, so you can kind of get a little compare and contrast as to what's happening. Here's the attachment stage, where the virus attaches to receptors on the cell membrane. That leads us into the entry stage, where the DNA or the RNA of the virus enters the host. After that, replication and assembly take place. Through the process of transcription and translation, the host cell is actually busily creating new proteins for the virus. And once those proteins are created, they are assembled, which leads us into step number 5, known as release. All those little viruses are released, and each one of those viruses will then be spread throughout the environment looking for a new host to repeat this cycle. So again, that's a little summary of the Lytic cycle. For those of you in my biology class, here is again the Kolbe quiz. Just a little quiz to see how well you think you understand the topic from today. All of these questions should be answered and found within the notes that we just took. If you want to write your answers on paper and then bring them to me before or after class, I would be happy to check your answers in class. But I do not want to put up the answers, because if I just give you the questions and then you all of a sudden have the answers on the next PowerPoint slide, something tells me many of you aren't going to take the time to look up the answers yourself. You're just going to flip and see what I've written for the answer. So I actually think the process and the act of looking up the answers yourself is an invaluable way to study. So if you do write the answers down, I'd be happy to check your answers for accuracy. So I hope you understood what we talked about, and if not, please come and see me.